Did you know that you have a self-destruct button? Okay, I'll stop with the puns. But in all actuality, we do. Introducing programmed cell death. But what is programmed cell death? Well, there are two types, necrosis and apoptosis. But for this video, we're mainly going to focus on apoptosis. So how does it work? Well, have you ever noticed that when you pop a bubble, it actually forms smaller bubbles before they disappear? Well, the process of death is pretty much the same. After receiving specific signals, the cell's membrane begins to shrink until it has become this. This process is called blebbing. Its DNA and organelles are then chopped up into small fragments, which then split from the cell, forming smaller shapes called apoptotic bodies. Macrophages then come in and eats these apoptotic bodies and basically cleans up the mess.
Yum.
So how is this any different from cell division? Well, as the great Nobel winner H. Robert Horwitz had once said, cell division forms the clay, whereas cell death sculpts the clay into its desired form. So those fingies that you have there, that was all because of apoptosis. But that's not all that it can do. While the process of cell death is the same, there are several ways in which this process can be initiated. Through the intrinsic and through the extrinsic pathways. The intrinsic pathway, also known as the mitochondrial pathway, sends signals from within the cell to begin the process of mitochondrial outer membrane permeabilization, which is controlled by a family of molecules called BCL2. Now, what does that mean? In other words, these molecules create openings within the mitochondria to release a protein called cytochrome C, which then attaches to other proteins to form apoptosomes that trigger enzymes called caspase that allows the cell to break down in an orderly fashion and eventually start the process of cell death. The extrinsic pathway, on the other hand, receives signals from other cells. When a death receptor binds with ligands or molecules from other cells, the cell then activates its death domain, which then calls for adapter proteins, which transmits the signal from the ligand to the inside of the cell. This causes extra structures called procaspase to bind with these adapter proteins and activate other enzymes, or caspases, to begin the process of apoptosis. The discovery of these two pathways are crucial in finding a cure for cancer and other viruses like our wonderful friend SARS-CoV-2 or COVID-19. By going deeper into the science behind signaling, we are able to find ways to specifically target cancerous or virus-infected cells and initiate apoptosis. Meaning one day, we might live in a world where we don't need chemo or Advil.